Hi everyone, this is Liam Sanyo from Inside Scientific, your favorite online source for life science webinars, virtual events, interviews, and educational content helping you do your best work. In this special episode of Industry Insights, I'm speaking with Dr. Rajendra Kumari and Dr. Gera Goverse, respectively the Executive Director of Integrated Solutions and Director of Immuno-Oncology at Crown Bioscience. A few days ago, the three of us ran a webinar about the cancer immunity cycle that covered various in vivo, in vitro, and ex vivo methods and technologies that can be used for preclinical immuno-oncology research. If you missed the webinar, the link will be in the description or on the page here, but today we're going to be taking a deeper dive into some of the concepts that Dr. Kumari and Dr. Goverse talked about in the webinar, including 2D versus 3D cell cultures, organoids, and high-content imaging. Alrighty then, Jen and Gera, thanks so much for joining us today. Are you ready to answer a few questions here?
Definitely.
Yes, we are.
Awesome. So just to recap, Jen, maybe you can lead this question, but what is the cancer immunity cycle?
Okay, thanks. So the cancer immunity cycle basically describes the necessary steps needed for our immune system to effectively eradicate cancer. There's a lot of genetic alterations within cancer. So these generate tumor-specific antigens that are very distinct from the normal tissue. And this is sufficient to trigger a cascade of biological events, starting with the presentation of those novel unique antigens on the cancer cells to the T cells. And then this results in the activation of those T cells so that when they reach the tumor microenvironment, they can recognize the tumor cells and effectively kill them. And this then releases more antigens and the whole cycle starts again. So this is very highly regulated process, and it prevents actually the recognition of self and autoimmunity. So there are numerous checkpoints. So you've got the PD-L1, CTL4. You've got a range of different stimulators. So CD40, OX40, CD137, and then many different types of inhibitors such as cytokines. And these work to keep that balance. But this also means that there's numerous points within that cycle that can be impaired or dysregulated, and this results in those tumours escaping that immune surveillance, and then you get progression of the disease. So this cycle captures very nicely, I think, the potential therapeutic targets that can be modulated to get that cycle back on track, and as well as help us determine why there may be some therapeutics that are not working. So I think in summary that cancer immunity cycle really describes the biology very well, but also how it can be very ineffective in cancer, so allowing that progression, and then also how immunotherapeutics can be effective or ineffective.
Yeah, definitely. And yesterday we were talking a whole bunch about, the different methods and technologies. And so obviously Crown Bioscience is really great at assessing a bunch of these steps along the cancer immunity cycle. But Jen, maybe you could share some of these ways that we were talking about yesterday. So how can you assess the steps of the cancer immunity cycle?
There's many different assays and model systems, and it's really hard to kind of describe everything. But generally you can model that cancer immunity cycle, or parts of it in vitro and in vivo. And this, I think, is going to be dependent on the biology of the target and the therapeutic being assessed. So you have to kind of design your assays and your investigations based on those key points. So I can start with in vivo, and Gera, if you want to jump in for in vitro. When we are using in vivo systems, for example, then ideally a fully functional immune system would allow the whole cancer immunity cycle to be assessed. And so the endpoint would be tumor killing to demonstrate efficacy. And this can include gem models where tumors arise as a result of engineered oncogenic drivers, or using the syngeneic systems or homograph systems. And these models, so the tumor develops over time with these in these models, and so they can then grow and avoid immune surveillance. So any successful therapeutic intervention will result in that tumor reduction. And then we can take samples at different stages from that study, looking at the tumor infiltration, the cytokine release, to see whether that response has worked or why it might not have worked. I think the biggest limitation, I think we do have to address the limitations of these systems, is that we're looking at murine-based biology in these. So any hypotheses that we may generate from studying and using these models needs to be then translated into this sort of human system. So we do need to still have further investigation into humanized systems. And again, in vivo, this is possible, but again, very complicated and limiting, very time consuming. So we have to have alternatives. I think this is where the in vitro systems really come in their own. And we can do in vitro human systems as well. So you can overcome some of those shortfalls, especially like the organoid system or the EVPT and really kind of break down the cancer immunity cycle so that you can look at the different stages as well.
Yeah, I totally agree with you, Jen. So indeed, as you mentioned, the breakdown in vitro assays, so really the cancer immunity cycle can be, you can really analyze more specific parts of the cycle with in vitro assays instead of just looking at the overall complexity of the whole in vivo system. So thereby it has, of course, advantages compared to in vitro systems. But then on the other hand, in vitro systems does allow you to work with the human system. You can incorporate the immune compartment in a maybe better setup than within some of the mice models. And in addition to that, the in vitro models, you can do much more of a higher throughput screening, right? So if you would like to test combinational effects, or you want to test different doses of your compounds to also find the best way of your efficacy of your compounds, that's really something that can be addressed in the in vitro systems. And it can be done in 2D cell cultures, but then those are just really high throughput and can give you some simple answers, rather say. But then if you go to a more complex system or more relevant methods using those 3D cell cultures, incorporating indeed organoids, Jen mentioned, or patient tissue, that would really be bringing you also to a higher value in actually the translation to the clinics. So in vitro assays can really bring a step closer to clinical trials, to successes for clinical trials.
Yeah, and these systems really work in concert, don't they? They really do help support. So you can go back and forth as well, and dive deeper into the sort of different sort of questions. I mean, certainly, I think the combinations is a very good example, because you just can't simulate all the different combinations in vivo as effectively as you can in vitro. So therefore, I think the in vitro really does help reduce the number of animals we're using in the end, and then also help us decide which ones to take forward and which to investigate further.
Yeah, that's a great point. I mean, you're right about them working in concert. Nobody's only, studying, nobody's only using in vitro models or only in vivo models. You really need both to get the full picture. I mean, you won't get high enough throughput or good enough scalability if you're only using in vivo models. And in vitro models just maybe aren't quite physiologically relevant enough to capture everything that you might get in an in vivo model. That's a really good point.
Yeah, I think some of the technologies, the advances in technologies, such as the 3D modeling, has taken it a step closer to being more relevant. So I think that's a really key feature of, as technology and techniques improve and get better, we get better at modeling cancer.
Yeah, great point. And we can talk about that now. So about organoids and high content imaging. So maybe Gera, can you explain these a bit more and how these technologies are impacting the field?
Yeah, sure, definitely. And actually, it's maybe interesting to also notice that during our webinar, we had one of those poll questions, and also to understand actually the audience. And we noticed that quite some of the audience, almost 50% or a little bit less than 50%, were not yet familiar with organoids. So I think it was very good that we had that crowd on the call, because I think organoids could be really the future for 3D cell cultures. So organoids are patient-derived, so I think that's really a difference to understand compared to cell lines. But in addition to that, because they are They are not a cell line, so they are not, for example, immortalized. But it's also based on a stem cell stimulation of growth. So you, by giving the right growth factors, you're stimulating the stem cell growth, and thereby you are actually capturing the heterogeneity of the tissue. So those stem cells can grow into those different subset of cells, and thereby you are not just looking at this, what you have with a cell line, whereby you just have one homogeneous population, which can give you valuable input on that specific response, right, of those cells. But I think the heterogeneity in organoids is really bringing value of getting a better understanding in how indeed patients will be reacting to that. And in addition to that, the high content imaging platform that Crown actually offers, and I think also in the webinars we showed many of the techniques out there are making use of imaging-based technology, maybe a little bit of a different setup, but it's really showing that the imaging technology brings also more value because the imaging can really capture morphological differences. So it's not just a killing analysis if we look at a tumor tissue, but also in addition to just looking at tumor tissue, the addition of the immune cells, you can really analyze the relative positioning of immune cells towards tumor tissue. And this is something that can only be captured via this type of imaging. And thereby, I think that the high content imaging platform that we offer is really bringing value of making use of organoids on one hand, which are really patient-related material, and thereby have a high value to the clinics. But then also, using imaging technology in a high throughput platform to analyze, indeed, as we just mentioned, treatments, but also combination treatments in one big go. And maybe coming back to our previous point, it's indeed there will always be a combination of in vitro and in vivo assays. And what's maybe interesting to mention is that from the organoids, we also have, for example, patient-derived xenograft organoids. So meaning that we have the in vivo models, so the patient tissue has been used in vivo models, but from those we also have the organoids. So that really brings again, where we can actually work together. First, put a screening in organoid models, and then follow up with in vivo models on that.
Fantastic. So, Gera, in addition to the organoids, that you also mentioned the use of ex vivo patient tissues. Can you explain the difference in testing with patient-derived organoids in these?
Yeah, I think that's a very good question, right? So on one hand, we have organoids, which are patient-derived, and as just mentioned, they do consist out of a heterogeneous population of cells, and thereby can bring a higher value for preclinical testing. Whereas the ex vivo patient tissue platform is really fresh patient material coming in from the clinics, which can either be resection material, biopsy material, but also liquids, for example, ascites or pleural infusions, for example. And those materials can be directly really used fresh. And I think that's really important that that material is not pre-cultured. There's no specific, because from that material you can also actually make organoids on one end, which could be interesting, and thereby you can set up a more stable type of organoid line, which does allow you to follow up, let's say, with a follow-up experiment. But the patient material really contains its native tumor microenvironment. And that's really the difference, right? So the organoids, you really stimulate the growth of the stem cells, but thereby you do lose, for example, other cellular players in those cultures, for example, stromal cells or immune cells. And in those patient tissues, you really maintain this native tumor microenvironment. And I think thereby the complexity of the tissue is maybe much higher, but it's almost, you are doing in vitro clinical trials in a petri dish, in a high throughput platform. So instead of using a clinical trial with getting as much as patients that you want in a patient, but then you can only test one treatment effect, right? So within the EVT type of platform, you can actually test multiple, again, treatments, combination treatments within one patient. So we call it kind of in vitro clinical trial, which is really valuable, again, for later translation to the clinics.
Yeah, certainly. So in response to one of the poll questions, the audience were using 2D cell culture for their in vitro research. And some were still unfamiliar with organoids. But do you think there's been a shift to using more 3D cultures?
I think actually it's interesting, right? So if you look at the poll, I think again it's about 50 to 50%. 50% is still working with 2D cell cultures, or at least indicated that they are working with 2D cell cultures. And maybe I'm a little bit biased working in a 3D in vitro type of platform at Crown Bioscience. So my last couple of scientific career have been always in 3D cell cultures. But on the other hand, it's interesting to see that on the other hand, 50% is already working with 3D cell cultures. So even 3D cell cultures in its own have been on the market for a bit of a longer time. I think the organoids have been introduced about a decade ago, but really since then have been shown in literature as well. There has been a much increase in the number of publications on organoids and really showing the clinical relevance of making use of those organoids. And therefore, I really think that it might be still hand in hand together with the in vivo models and with other 2D, maybe in vitro based assays. But I think it's really, where everyone wants to work with. On one hand, you can perform or analyze more treatments again, combinational treatments in those type of 3D assays with organoids. So you are working in a relevant system. You can make it as complex as you want by incorporating different immune cells, different other cellular players, so to kind of really reconstitute the tumor microenvironment. But then still allow you this kind of screening based platform. So I really think that this will be where everyone in the end will need to, will be moving forward in this 3D space. How do you see that, Jen?
Yeah, no, I think very good points there, Hera. I think there has. I mean, certainly, I've seen shifts away from 2D cell culture. I mean, there's a lot of criticism of cells that are grown in 2D. Even when you move them into xenograft systems, they form cancers which don't represent the original cancer type, and that is partly due to their adaptation to 2D systems. So I think the 3D takes those cells and puts them into more physiological relevant types of environment, which means that they remember their origins, and so they do represent the tumor in the end far more faithfully than the 2D cells. And we've seen collections like the NCI-60 panel being retired in favor of more clinically relevant systems, and certainly that is where patient-derived systems are coming through. And I think the challenges that we've had with patient tissue, the access to tissue, the use of tissue, these systems like organoids, like the EVPT, really make them their use much more efficient, cost-effective, and translationally valuable. So I think there will be more adoption, and we've seen more and more people certainly asking us for these types of systems.
Yeah, definitely. I mean, I'm sure we all know that there's just an enormous number of agents that were pursued that get dropped during clinical trials. And I think part of that is because they're not really caught early enough that they won't quite work out. Of course, there's a lot of reasons for that. But if we can use more physiological relevant models sooner, then maybe we can catch some of these and don't spend quite as much money developing agents that will never really work out. But yeah, that's a really, really good point.
I think actually, Liam, that's a very important point, right? So although there's not so much of publication on that, but we do know that there are indeed compounds that went to clinical trials that were showing really good results, let's say, on preclinical testing, which was done on 2D cell cultures in vitro, also in vivo in mice studies. But that indeed, you kind of lack the human 3D physiologically in those settings. And then it turns out when they go to clinical trial, they might really just fail. And later on, they also have, in some of those examples, shown that when they were performing those studies in 3D cultures, that actually indeed genes that were expressed in 3D cell cultures, which were not expressed in 2D cell cultures, were actually the trigger that why some of those molecules were not working. So I think that's a very good point. And although literature is not showing them all, but there's definitely some failure in clinical trial because we lack a certain expression or a sensitivity by not making use of the right preclinical model.
Yeah, it's really interesting, the kind of things that 3D culture can capture that 2D hasn't been able to. It's kind of an exciting time to be in immuno-oncology for sure. So shifting gears a bit to maybe a more general question, and Jen, maybe you can lead this one, but why should drug developers work with Crown Bioscience?
Yeah, I mean, we offer, as you've heard, a broad range of capabilities for oncology and immuno-oncology, and this includes biomarkers and bioinformatics, which we probably haven't touched a huge amount on. We have a long-standing track record in working with drug developers across pharma and biotech for these preclinical studies and investigating sort of numerous different types of therapeutics, not just immune checkpoint inhibitors, but cellular therapies, oncolytic viruses, vaccines, gene therapies, microbiome modulators, as well as looking at resistant mechanisms and those combination strategies that we talked about. And we also demonstrate that of this sort of experience and capability by sharing the data that we've generated and how we've characterized our models. And you can access through this through our website and our databases. So there's a lot of sort of molecular features on our patient-derived models. We've got the clinical annotation and any relevant data. And I think as highlighted already, those patient-derived systems for in vitro, in vivo and ex vivo is really a unique capability. And the ability to then run those clinical trial-like studies in those different platforms, again, I think is making a big impact and certainly an area that we're very, committed to developing out further. So I think there's a lot of capabilities that we can provide, a lot of expertise and guide which platforms and systems are best for a therapeutic or a stage of the drug.
Yeah, and maybe just to add on that, I think indeed different stages of the drug and a different type of drugs, but again, bringing the synergy also of the in vitro versus the in vivo models that we can actually nicely follow up with those models. So we do have experts in both fields, and thereby, we can really start from the beginning. And I think in the webinar, we gave one of those examples where we were involved, let's say, in the beginning of identifying hit compounds, and all the way in the end that led to a compound that went into clinical trials in one of our projects with our collaborators. I think that's really showing that we are not just a part of the cancer immunity cycle that you can address, and also, so it's really the full package, right? So from the beginning to the end, I think that's really the value.
Fantastic. Yeah, great arguments made. All right, so I think just for the last question here, so if somebody wanted to learn more, if somebody wanted to talk with somebody at Crown Bioscience, where should they go, or what should they do?
I think you can start with the website, so www.crownbio.com. There's a whole range of resources there. So if you need more information, there's fact sheets, there's white papers. We put our publications on there as well as presentations. So you can really get a lot more detailed information just accessing those resources. And then if you need to ask any questions, you can just contact us via the webpage as well. And the databases as well, you can follow the link on there, register for them. And then there's like different types. So for organoids, for example, there's the organoid base, and then we've got ones for PDX models, for our murine systems, and then for our cell line models. So I highly recommend visiting that website and reaching out to us as soon as you have a question.
Perfect. Well, Jen and Gera, thanks again so much for joining us today. It's been really great talking with you.
Thank you so much.
Thanks a lot. That's been great.
We hope you enjoyed this special episode of Industry Insights. If you did, then stay tuned. We'll be making 2 more episodes with Crown Bioscience in the coming weeks, covering different methods, challenges, and opportunities in immuno-oncology. Thanks everyone for listening, and don't forget to subscribe.